Good evening. Thank you for joining us. Welcome to the second in a series of 5 Florida Keys Mosquito Control District Oxytec public educational webinars. My name is Meredith Fensom. I'm Oxytec's Head of Public Affairs and a Florida native. Joining us are Dr. Neil Morrison. Neil has a PhD in entomology from the University of Oxford, and he is Oxytec's Head of Agriculture Programs. We also have Dr. Nathan Rose. Nathan has a PhD in chemical biology, also from the University of Oxford, and he is the head of Oxytec's regulatory affairs. Many of you in the Florida Keys have met Nathan. Before starting this webinar, we'd like to quickly review with you the others coming up in this series. Today is Lab to Field. Next Tuesday, August the 11th, same time, we'll review Oxytec's, next Tuesday, August 4th, same time, we will review our community engagement plans with Florida Keys Mosquito Control District and Oxytec's past work in this area. The following Tuesday, August the 11th, also at 5 p.m. Eastern, we'll review Oxytec's vector control performance. Then finally, on Monday, August 17th, also 5 p.m. Eastern, we will review assessment, oversight, and validation of Oxytec's technology. All of these webinars are open to everyone. They are all recorded and made available after the event. We'll answer as many questions as we can live. We may group similar questions together. If time runs out, we will accept questions in writing via florida@oxytec.com. We'll also publish the questions asked and answered after the event, and we'll provide external validation or resources. This evening, we have an hour for the webinar. We'll use the first half hour for a presentation, and then we'll use the second half for questions and answer. You all can begin to submit your questions now through the chat, the Q&A function. Just write your question, remember to press send. We will not be responding to you by text. We will respond to you vocally during the live webinar. And again, if we run out of time for questions at the end, you can submit them to us by email, florida@oxytec.com, and we will reply to you. Now we'll turn it over to Neil.
Thanks, Meredith. Just a quick introduction to Oxytec, and when we were founded back in 2002 out of science developed at Oxford University. Since when we have built out and validated our technology platform with the help of strong partnerships with independent world-leading experts and organizations around the world. So we're passionate about our mission to deliver impact to human health and agriculture through delivering biological solutions for management of pests that transmit diseases to people and destroy crops. So you can find a lot of the detail of our science in over 100 peer-reviewed publications, which by definition are reviewed by world leaders in their field. And so with this independent validation, you can really find information that describes essentially the effectiveness of our insects, the sustainability of our insects and the safety of our insects. And these papers are available for all to see. So wherever we work in the world to deliver impact where it's needed most, we go through another level of scrutiny and validation, and that's government bodies and independent regulators in places like the US for this mosquito project. This project has been reviewed and approved by not only the Environmental Protection Agency, but also multiple state-level regulators who have found that this project will be safe for people and the environment. And the same applies in all the countries where we work, not only in the US. So, for example, in Brazil, where we've made significant strides with our mosquitoes, again, all of our projects are scrutinized and validated by independent experts in government bodies. So just a little background on why we've developed our mosquito. Aedes aegypti is the primary vector of diseases like dengue and Zika that threaten human health around the world. And it's a very difficult mosquito to control. And current management tools are under heavy pressure. So new tools are sorely needed to manage this vector. And we've developed a solution that's proven to be highly effective at reducing Aedes aegypti populations in urban environments. Its mode of action is mating based between a male and a female mosquito. So its effect is very species specific. And so it's harmless to the wider ecosystem. The mosquitoes that we release into the environment are safe and non-toxic, so pose no threat to predators that might eat them. And we also don't release females. Remember, it's the female mosquito that bites. The males can't bite people. We don't use tetracycline in our deployments, and our approach is highly suitable for integrated vector management programs, and will be a cost-effective tool in vector control authorities' toolboxes. So before OX5034, there was our first generation Aedes aegypti, with which we released around 1 billion adults in deployments from Brazil to the Cayman Islands, Panama, and Malaysia. And these deployments demonstrated outstanding levels of suppression of the Aedes aegypti mosquito, with no reports of harm to people or the environment. The second generation OX5034 mosquito that we aim to trial to deploy in Florida brings more advantages to the table. So we've demonstrated its effectiveness in multiple urban deployments in Brazil, and it offers a key advantage in that it has built-in genetic sexing, so we can't release biting females of this 5034 strain. So not only does that result in no biting mosquitoes in the environment, but it also opens up the opportunity to deploy at all life stages, essentially. For example, eggs. Any eggs that we deploy in the field will be male only when they emerge as adults. So this 5034 mosquito offers highly targeted suppression. Again, it's males, our males mating with wild females. So the effect is highly species specific. The two proteins we introduce into the insect are safe, they're non-toxic, non-allergenic, so they don't pose a threat to any predators that feed on the mosquitoes in the environment. We've proven the effectiveness in deployments in Brazil, and the male-only releases mean we don't release females into the environment. We also introduce a marker gene into our mosquitoes, so that allows us to trace our mosquitoes in the environment. And the gene means that the insects are self-limiting in the environment. So after we stop releasing, the gene disappears from the mosquito population within a few generations. So we leave no permanent footprint in the local mosquito population. So before we even get to the field, our insects undergo multiple years of research and characterization. Right from initial development of the strain in the lab, we conduct characterization of the insects, detailed safety studies, and studies to demonstrate effectiveness. And with this, we're able to populate data packages that we submit to regulatory bodies. And we have a great track record in regulatory approvals around the world. And that's enabled us to conduct field trials to demonstrate efficacy of our insects out there in the field. Now, before we're able to deploy a more commercial setting, our mosquitoes will undergo further regulatory assessment and validation. And then after, once we have that, and that's not the end of the story, we take stewardship of our products extremely seriously. And so there will be an ongoing and continual process of quality control and safety controls to ensure that we continue to deliver a product that's safe and effective for the purpose in the field. I'll hand over to Nathan here, who'll go into a little bit more detail of the science and report on some of the proven success we've had with 5034 out there in the field.
Thanks, Neil. So what I'd like to do in the next few slides is just to talk through some of these steps that Neil has just outlined, from the initial development of the 5034 strain through some of the validation, the regulatory assessments, and then some of the field studies that we have done in Brazil. So the way we started by developing 5034 was to introduce two genes into the Aedes aegypti mosquito. These two genes are widely used, and they're very well studied in both cases for more than 20 years. So these two genes, the first is a male-selecting self-limiting gene, which produces a protein called TTAV. And the second is a fluorescent marker gene, which produces a fluorescent protein called DS-Red. And the way that these genes are introduced into the mosquito is shown here. The DNA which contains these two genes is injected into Aedes aegypti eggs using extremely fine needles, as you can see there. Those eggs are hatched, and any mosquitoes which have actually incorporated those genes into their own DNA are then selected by looking for that fluorescent marker protein in those mosquitoes. And then once we've identified those, then we will be able to actually keep on breeding those mosquitoes in the lab without the need to continually inject DNA into them. So a little bit more information about these two genes. The first is the self-limiting gene, which produces the TTAV protein. This protein is, as I said, widely used in biology. It's non-toxic and non-allergenic. It's made in 5034 female mosquitoes, and it stops female mosquitoes from being able to survive to adulthood. Male mosquitoes are unaffected, and this means that we can produce male-only batches of mosquitoes, either in a production facility or, as we plan in this project in Florida, using egg release devices. And I'll go into a little more detail about those on the coming slides. And the other important aspect of the self-limiting gene is that this is what helps to suppress wild mosquito populations. Because none of the female offspring that inherit this gene can survive, we're able to actually suppress those populations substantially by releasing the male mosquitoes into the wild. We have released about 20 million male OX5034 mosquitoes in Brazil over the last 2 years, and 0 females at the same time. And this is because of the very high effectiveness of the self-limiting gene. The second gene which we've introduced into the mosquito is a fluorescent marker gene, and this produces a protein called DS-Red2. This is also a non-toxic and non-allergenic protein, which has been studied for about 20 years. It is widely used in biology, and it is visible under special lights and special filters. And under those special filters, you can see the mosquitoes glowing bright orange, which allows us to identify them and differentiate them from wild mosquitoes. It's produced in mosquitoes at all life stages, so we can see this whether they are larvae or pupae or adults. And it can also be passed on by the male mosquitoes, which are released to their offspring, and that allows us to track these mosquitoes in the field. Several questions have come up in the last couple of public webinars about something called introgression, and I'd just like to address that here in a little bit more detail. So in addition to these two genes which we've put into the Aedes aegypti mosquito, the mosquito has many thousands of natural genes which are in the mosquito. And those could be called the background genes or the background genetics of that mosquito. And because the male mosquitoes which are released into the field will pass on not just the two genes that we've introduced, but also the background genes to their offspring. Their female offspring will die, but their male offspring will survive, and they will then be able to continue to pass on some of those background genes into the wild population. And that's something known as introgression. And this is something that has been specifically reviewed by the EPA, by the CDC, and also by the Florida state regulatory authorities, and then also by the regulators in Brazil. And they concluded, looking at a lot of data on this particular issue, that there was no added risk to human health or the environment as a result of these background genes being passed on to the wild mosquitoes. They also confirmed that there was no risk of the wild mosquitoes becoming more vigorous or more fit, something called hybrid vigor. There is also no risk of the wild mosquitoes becoming more resistant to insecticides. In fact, the opposite is true, because the background genes in the mosquitoes which we release make those mosquitoes susceptible or sensitive to insecticides. And that means that if they pass those genes on to the wild ones, they'll make the wild ones also sensitive to insecticides. And that's a really important aspect of these mosquitoes. And they also confirmed, studying a lot of data from a lot of independent scientists, that there was no risk of these making the wild mosquitoes any better at transmitting disease. And that's something called vectorial capacity or vector competence. So this is something which has been very carefully and heavily studied by the regulatory authorities and also by the US health authorities when it comes to assessing this particular project. So in addition to that, the regulatory authorities carried out an in-depth review of a lot of data generated by Oxytec, but also generated by independent scientists, and also a lot of data that's already present in the scientific literature. I won't go into a lot of detail here, but in total, probably over 4,500 pages of data reviewed by the EPA, including 2,500 pages of scientific peer-reviewed literature. And the data requirements which were fulfilled by Oxytec covered aspects of environmental assessments, which I'll talk about a little bit more on the next slide, human health assessments, and then detailed characterization of the OX5034 mosquito strain and its performance. In addition, I just want to talk a little bit more about the environmental assessments that were done by the EPA and also by the Florida state authorities. They looked at the environment very broadly speaking, so looking at animals, plants, any endangered species, any aspects of critical habitat present in the Florida Keys, and determined no effects on endangered species or critical habitat, whether direct or indirect. So a direct effect would be something eating a mosquito. An indirect effect might be what happens if you reduce the mosquito population, what happens to the ecosystem. So the EPA looked at the effects on fish, birds, mammals, plants, other invertebrates, other aquatic animals, things like amphibians. They determined in the course of their evaluations that Aedes aegypti is a negligible part of bird, amphibian, or bat diets. They just don't rely on Aedes aegypti as an important food source. And this means that if you're reducing the population of this mosquito, you will have negligible impact on any animal that might eat it. In addition, they confirmed that the 5034 male mosquitoes cannot bite, which means they cannot bite people, and they also cannot bite wildlife. In addition, they looked at direct effects, for example, experiments done by third-party independent labs, looking at what happens when fish or invertebrates eat an OX5034 mosquito larva, and showing that fish or invertebrates subsisting on a diet of 70% mosquito larvae fared no differently to fish or invertebrates which were fed non-genetically modified mosquito larvae. So again, confirming the excellent safety profile of this mosquito as it relates to the environment. I'd like to talk a little about what happens when we actually make these mosquitoes, and I'll talk a little bit about the role of tetracycline in that process as well. So in the United Kingdom, we produce OX5034 mosquito eggs, which are then shipped to the United States or anywhere else that we might want to be doing field deployments. And then those eggs can be used in the field, hatched in the field to produce male mosquitoes only. However, to produce eggs, we need to have male and female mosquitoes breeding, and then female mosquitoes laying eggs. And to be able to do that, we need to add a small amount of tetracycline to the water that the male and female larval mosquitoes are living in and growing in. And that enables the female mosquitoes to survive to adulthood. After the larvae turn into pupae, they are washed and moved to clean water. The pupae are then put into cages where they close into adult mosquitoes, males and females. They will mate, and then the females will lay eggs. And then those eggs are surface sterilized with disinfectant, which is far stronger than hospital-grade disinfectant. And then they are packaged into capsules and can be shipped. At all stages, there is very strict quality control, partly to ensure an effective and good quality product, but also to ensure that we are compliant with all of the regulations put in place by the EPA as they regulate this particular project. Just quickly to talk about tetracycline, as I've said, it is only used to help female larvae survive through to adulthood. That means that it's only necessary to be used in the United Kingdom where we are producing eggs. It is not used in the United States where we would actually be hatching these eggs out to male adult mosquitoes in the field. And the total amount of tetracycline used to produce all of the eggs required for the whole project planned for Florida would be less than 5 grams. And comparison, packet of sugar weighs approximately 5 grams. That's the total amount of tetracycline that would actually be required in the United Kingdom for this particular project. What happens next? So the eggs are then shipped once they've been packaged to, in this case, let's say Florida. And then we would take those eggs, we would put them into what we call a mosquito rearing box. And in that box, the eggs will have water added and also some diet added. And they'll hatch there. There's no tetracycline in that box, which means that all of the females will die, but all of the males will survive to adulthood, and then they can fly out of that box, go out into the field, mate with wild females, and pass on those 2 genes that we talked about earlier. So in that box, we're only producing male adults. There's no tetracycline in the box and no tetracycline usage in Florida at all. And in the field, these boxes will be placed, assuming that we proceed with this project, by FKMCD or Oxytec operators in out-of-the-way areas where those male mosquitoes can hatch and develop in peace. One more brief comment on tetracycline. As I've said, the eggs that have been shipped to Florida would never have been in contact with tetracycline, and it is used only in the United Kingdom. And this is something which EPA has assessed in great detail. And having looked at this, they concluded there's negligible risk that the use of the 5034 mosquitoes would spread antibiotic-resistant bacteria in the US environment. And more details of their assessment and their reasoning process can be found in all of the risk assessment documents that have been published by the EPA. Just for comparison as well, because I think this is a useful contrast to draw, the amount of tetracycline used, as I've said, is less than 5 grams. That's the equivalent of about 2 human therapeutic courses of antibiotic. In the US every year, doctors and hospitals prescribe, it's reckoned, about 47 million antibiotic courses for infections that don't actually need antibiotics, and that's CDC data. In Florida as well, we know that a large amount of tetracyclines and other antibiotics are approved for spraying each year onto particularly citrus farms. And the amount that's currently authorized is up to 388,000 pounds of antibiotic each year on citrus. So just, I think it's worth contrasting that with the amount of tetracycline that will be used in the United Kingdom and seeing there's roughly an 88 million time difference between the amount that we use for this project compared to what's already in use in Florida. All right. So we've talked about how the mosquitoes are produced. We've talked about how the strain was made originally. We've talked about some of the data that's submitted to regulators to assess this. I want to briefly talk about what the mosquito has actually done in the field before we finish up this presentation. So Oxytec has more than a decade of mosquito deployments in different parts of the world, starting in the Cayman Islands and in Brazil, some projects in Panama and Malaysia as well, and then larger scale deployments in Brazil with our first generation mosquito, OX513A. We have done 2 years of projects with the OX5034 mosquito in Brazil. The first one was a pilot in 2018 to 2019 with extremely effective suppression, 96% peak suppression of the wild population in the city where that was deployed. And then within the last few months, a trial of the mosquito rearing box that I talked about a few moments ago, also in Brazil. As part of all of those projects, we carry out extensive community engagement, and that's what we're doing right now. And we will continue to do this together with FKMCD to engage and to listen and to answer questions and to explain why this project's important, how it works, and why it's safe. And just briefly to talk about the data that have come from Brazil from this mosquito rearing box deployment of adult male mosquitoes in the field. This worked very well, and it gave us suppression of the wild population far quicker than with first generation mosquitoes. On the right-hand side of the slide, you can see a graph showing the reduction in abundance of the wild mosquitoes in the dotted line, reaching a weekly high of over 90%. This was about 6 weeks faster than we would have been able to achieve with the first generation mosquito. And this validates all of the things that Neil talked about earlier, the reasons for developing the second generation mosquito OX5034. Also, as part of that trial, safe, no unintended impacts, male-only release was achieved, not a single female released. It's fully self-limiting, did not persist in the wild after release stopped. And we had very strong suppression and also very high public acceptance throughout that project. Briefly, we also continue to work with independent scientists and reviewers, especially as we plan this project moving forward. And in addition to the advisory board that we talked about at our last public webinar, we also have collaboration with the CDC that will be part of this project, helping to assist and to evaluate the data so that we can make sure that we have very robust data evaluation as we take that back to the regulators. Okay, one last thing just to quickly address, and this is a question that we often get. Do our mosquitoes bite? As I've said, we are only releasing male mosquitoes throughout this project, and the self-limiting gene stops any female mosquitoes from being able to develop or be able to be released. Male mosquitoes cannot bite. They do not have the mouth parts to do so, only females do. And so there is absolutely no risk of being bitten by an Oxytec mosquito throughout this particular project. Okay, I'm going to hand this back to Neil, just to quickly finish this up.
Thanks, Nathan. So before we finish up, I'll just address some of the frequently answered questions that we receive, and then we'll open the floor to questions from the audience. So firstly, if approved, when would this project take place? In 2021. So we recognize the gravity of the COVID-19 situation in Florida and elsewhere, and so our focus is entirely on initiating the project, if approved, in 2021. Our human positive proposed project? No, our focus is entirely on the performance of our mosquitoes that are being released into the trial sites in Florida Keys. Nathan touched on impact on the environment. Will they be harm to the unique ecosystem in the Florida Keys? No, our insects are safe, they're non-toxic, and this is certainly validated by the EPA and Florida state regulators in their approval of the project from a regulatory perspective. And that's supported by the science that we have carried out over multiple years to validate that. Nathan went into some detail about tetracycline. Again, to repeat, we won't be using tetracycline as part of this project in Florida. And I'll hand back to Meredith.
Thank you, Neil and Nathan. We've gotten a lot of great questions, and I'll turn to those in a moment, and I'll be answering the first one. But first wanted to mention to everyone viewing tonight, we're not going to identify who has asked the questions. Different from our webinar a couple of weeks ago. We want people to feel just completely free and comfortable to ask any question that they are curious about. So we will not be identifying who is asking these questions, and we want you all to feel very free and comfortable to ask anything you're curious about. Our first question is this webinar just Oxytec people talking, or can we ask board members questions? And I think this question is referring to the Board of Commissioners for the Florida Keys Mosquito Control District. And we had a public educational webinar co-hosted with the Florida Keys Mosquito Control District 2 weeks ago. It was Tuesday, July 14th. It was 3 hours long. And we did have members of the FKMCD team as panelists, and they did respond to questions. They joined us in answering questions for about 2.5 hours that evening. Last Tuesday, on July 21st, the FKMCD Board of Commissioners had their monthly board meeting, and there was an Oxytec component. I believe it lasted for about 2.5 hours, and that was largely an opportunity for members of the public to make their statements about Oxytec and the proposed project. Tonight, the focus is lab to field, and this is a big focus on Oxytec's technology this evening. So no, the board members, the commissioners of the Florida Keys Mosquito Control District will not be responding to questions tonight. We have Oxytec here to respond to questions for you. And I'll note that for our webinar next Tuesday about community engagement, the Florida Keys Mosquito Control District will be a part of that webinar, and they will be answering questions then. And the community engagement is something that, very much involves them. So, okay, the next question, and I believe we can send this one to Nathan, says tetracycline is controversial here in the Keys. What did the regulators decide?
So I talked through some of that in the slides which I showed earlier, and the EPA looked at this question in quite a lot of detail. They looked at the production process, they looked at where tetracycline is used, and as I've explained, that's only in the United Kingdom, and they assessed very carefully whether there was any risk of tetracycline-resistant bacteria being spread by male mosquitoes which are released. And because of the production process, because of the fact that the eggs are never in contact with tetracycline, that the male mosquitoes that are actually released are never in contact with tetracycline, they concluded that there was no risk of spreading tetracycline-resistant bacteria in the environment. And the full details of their reasoning for that, the independent science which they cited in that conclusion, that's all available on the EPA's website. And I think we've also put it on our website as part of the information available about this particular project.
Thank you, Nathan. Our next question says, Oxytec's initiative depends on citizen support. If GM female mosquitoes are detected, will you shut down your trial or experiment?
So that's a good question. As I've explained, the self-limiting gene which we put into the mosquito means that you cannot have female mosquitoes being produced unless there is tetracycline present. One of the sort of requirements for this particular project imposed by the EPA is that if at any time we were to detect a female mosquito, we would have to immediately stop releases, and we would have to inform the regulators, both state and federal regulators, and take steps to spray any of other mosquitoes of ours which might be out in the field. So, as I've said, we don't expect that to be the case at all. based on extensive experiments and based on more than 2 years of precedent in Brazil, where we have not seen a single female mosquito. But if we were to find one, the regulators would expect us to stop the trial.
Thank you, Nathan. Our next question says, how does your mosquito disappear after a few generations if males survive to breed with wild females? And Nathan, this will go to you too. And the question again is, how does your mosquito disappear after a few generations if males survive, and then in parentheses to breed with wild females?
Mm-hmm. Okay, that's a good question. And the way to understand this is to think about how many copies of the gene are actually being passed on with each subsequent generation of the mosquito. So when we release the mosquitoes, we release mosquitoes that are male, which carry 2 copies of the self-limiting gene and 2 copies of the fluorescent gene. They pass that on to all of their offspring, which means that every single one of their offspring inherits 1 copy of those genes. The females will die, the males will survive, and the males which survive will then only have 1 copy of that gene, which they can pass on to their offspring. So these are the grandchildren of the mosquitoes which were originally released. And those grandchildren, the sort of second generation down, only half of those will actually inherit a copy of that gene. The other half will not inherit any copies of the self-limiting gene or the fluorescent gene. If we go 1 generation below that, the great-grandchildren, it'll be only a quarter of that generation which will be able to inherit a copy of those 2 genes, and so on down. So it will dilute its way out of the wild population over time. At the same time, we expect the wild population to be decreasing substantially because we are stopping female offspring which inherit those genes from surviving. But what we have seen in both lab experiments and in Brazil in 2 years of field releases is that it behaves exactly as we expect, that within a few generations after we stop releasing mosquitoes, the self-limiting gene and the fluorescent gene are completely gone.
Nathan, if I could just summarize that in a sentence, I would say that any gene, whichever gene it is, if half of the offspring that carry that gene can't survive, then it's ultimately destined to decline to extinction over time. Mathematics just dictates that it will reduce by half in each generation at least.
Thank you, Neil. And we may have lost Nathan, but we'll move to the next question. If Nathan is able to join us, again, we'll give it to him. And otherwise, Neil, I'll ask for you to please answer this. The writer says, I am watching from Canada. Are there any discussions underway to deploy Oxytec mosquitoes in regions with mosquito-dense populations that do not carry serious disease? Not as serious an issue as in countries with Zika or malaria, but still a major nuisance.
Mm-hmm. I'm happy to take that if that's okay, Meredith.
Please.
I think there's, as the writer kind of notes, the need is most urgent for disease vectors, and that's really where we seek to deliver impact in the near and medium term. But there's absolutely no reason why we can't provide a solution for mosquitoes which cause nuisance through biting. We recognise that is a widespread and life-affecting problem for many people. For example, in Canada, we've looked at the potential mosquitoes that our technology platform could address, and certainly we wouldn't restrict ourselves to disease vectors if we felt that we could, provide positive impact to communities that are affected by other mosquitoes.
Thank you, Neil. And okay, we'll move to the next question. And Nathan, we'll hand this one to you, if that's okay. It is, in an earlier webinar, I believe the amount of tetracycline used to produce mosquitoes for the Keys would be less than a sugar packet. Today, you said there would be no tetracycline in deployments. Can you explain the role of tetracycline in this process?
Sure. So I'm just going to go back to one of the slides that I shared earlier, and I'm hoping that this will help to explain. Tetracycline will be used, and it'll be about 5 grams, less than a sugar packet, but it'll only be used in the United Kingdom where we produce mosquito eggs, and that's to help us to produce female mosquitoes to lay those eggs in the production facility in the UK. And those eggs are then shipped to wherever we're doing deployments, in this case, Florida. And then in Florida, those eggs are then put into these mosquito rearing boxes, which are placed in the field. And that's where those eggs will hatch and turn into male mosquitoes. So there is no tetracycline used in the deployment of the male mosquitoes, if that's in Florida or whether it's in Brazil or anywhere else we might be deploying these mosquitoes. So it is used, but only in the production of female mosquitoes, which will actually be producing those eggs. And those female mosquitoes only encounter that tetracycline when they are larvae. Once they become pupae, they no longer need the tetracycline, and those pupae or those adults never encounter it again. The eggs never encounter it. The eggs are surface sterilized, packed into capsules, and then shipped. And then once they hatch in the field in Florida, those male mosquitoes will never have encountered tetracycline either. So that's what I mean when I say there's no tetracycline in deployments. I mean in field deployments or in the production of the mosquitoes, which will actually be put into the field. So I hope that helps to answer that question.
Thank you, Nathan. We'll give you the next question too. It says, is there a way to reduce the lifespan of male Oxytec mosquitoes to a sweet spot where they mature enough to mate, but don't live long enough for introgressing to potentially be an issue?
Okay, so introgressing is passing on the background, the sort of wild genes from the mosquito into the wild population. And so the only way you can pass on that genes is through mating. So mating is what makes our mosquitoes effective because it enables them to pass on the self-limiting gene, which means that female mosquitoes will die. But mating is also what enables the background genes to be passed on into the wild population. And introgression is not an issue because it's something which has been assessed in great detail. And the assessments from the EPA and the CDC concluded there was no risk associated with introgression. So they looked at a range of things, but particularly looking at things like hybrid vigor, looking at things like insecticide resistance or susceptibility, and also vector competence. And for all of those things, they concluded there was no added risk by introgressing background genes into the wild population.
Thank you, Nathan. The next question is for you too. It says, if a female bred with OX5034, so our second generation mosquito, and laid eggs in an environment with tetracycline, could females survive?
Okay, I think it's a great question. So theoretically, if there was a high enough concentration of tetracycline in a breeding site for a female Aedes aegypti mosquito that had mated with the 5034-1, some females could survive. And so that's why the EPA looked at this particular question in great detail as well. So they looked at the likelihood of there being any tetracycline in the environment where these mosquitoes might be released. They looked at the concentrations of tetracycline which are needed to enable females to actually survive to adulthood. And looking at those two things together, they concluded there was no risk of this actually happening throughout the course of the trial, because environmental sources of tetracycline in the Florida Keys are not going to be, first of all, they didn't think that there would be significant environmental sources of tetracycline. And they also looked at particularly where these mosquitoes are breeding. And Aedes aegypti mosquitoes tend to breed in small containers, tires, flower pots, anything that you might have around your yard. And the likelihood of finding tetracycline in any of those sources is incredibly low, to zero. And for that reason, when they assessed any risk of this happening, they concluded that there was no risk.
Thank you, Nathan. The next question is for you too. It says, could you explain again, how do you make sure it is a female-only effect?
Okay, so this goes back to when we were actually developing this particular mosquito. This is one of the things that we are assessing as we create the strains that we want to actually take forward. So what we would do is we would look at what happens when you breed these mosquitoes in the absence of tetracycline. Are you able to get any female mosquitoes surviving in the absence of tetracycline? And the answer for the OX5034 strain was no. So we've done this many times with many thousands, if not hundreds of thousands of mosquitoes in the lab. And we've never seen a female mosquito actually coming all the way through to adulthood without tetracycline present. And we've also looked at this in the field in Brazil, where we've released, as I said, about 20 million male mosquitoes, and we've never seen a female mosquito in any of that production. And we continue as part of our quality control process for this particular mosquito to make sure that we are checking with every new generation of the mosquito that we're producing. By every generation, I mean every time we get females to produce a new batch of eggs, we will take some of these eggs and we will check them to make sure that we are not able to get any females surviving from those eggs as well. And that's part of the quality control system which is required by the EPA for this particular project.
Thank you, Nathan. The next question will go to you, Neil. And the question is, why doesn't Oxytec conduct the experiment in an area where there is no human population?
Okay, I think the crux of that answer is really down to the biology of the mosquito. So it's it is highly associated with humans. The wild mosquitoes, the females, they blood feed on people, they live around people's houses, they breed in people's yards, and they don't typically live in great numbers in rural environments away from where people live. And so to demonstrate performance of our mosquitoes in finding and mating with wild Aedes aegypti and to demonstrate that we're able to control them, urban or residential environments are really where we can do that. Yeah, so it's an anthropophilic mosquito. It's tied to human habitations, and therefore that's where we seek to do our work. I hope that answers the question.
Thank you, Neil. The next question is for you also. And the question is, are all of the peer papers available for the public to read? If so, where can we find them? Also, is there an index for easy reference by subject? And this person wanted to be identified as someone who is in Key Largo. Okay,
so a first stop would probably be the Oxitec website. So I think on our technology page on the website, we have a list of many of those publications. I think with links that you can access directly to those papers. In terms of availability to read, historically peer-reviewed papers have been behind a paywall. So it may not be that you can access those papers without paying for them. But increasingly, we are proactively seeking to publish all of our science in open access publications. And so certainly our most recent papers, you'll be able to access free of charge as we seek to just proactively provide that information in a transparent way. Sorry, Meredith, did I interrupt?
I was just going to add one thing, Neil, if I could there, just to say that so those papers are not sorted by subject, but if there's any particular subject that you want to know what we've published on that subject, just send an email to the Florida@oxytec.com email and we can point you to some of the relevant papers.
Yes, please. Okay. Neil, I think you answered the question. Is there more you wanted to say?
Well, only that if those sources of information don't satisfy, you can quite easily search for all of our papers. They should be available on, through internet searches by subject matter and including the term Oxitec in the search term.
Yeah, Google Scholar or PubMed are good places to search for academic papers.
Mm-hmm.
Thank you, Neil and Nathan. And again, to everyone watching, use the florida@oxytec.com email address. We respond to questions there every day. And if you're needing more information or would like some of these papers, please write to us and ask. We'll be responsive. The next question is for Nathan. And the question is, would the technology for creating the genetically modified Aedes aegypti theoretically function for other species as well, notably Anopheles gambii? Are there projects underway in Africa to address Anopheles gambii? I hope I'm pronouncing that correctly. And then the question also includes malaria.
Okay, so Anopheles gambii is one of the major vectors for malaria, particularly in West Africa and Central Africa. Yes, our technology can definitely work in Anopheles, and we have 2 projects underway right now which are funded by the Bill and Melinda Gates Foundation to develop this technology in Anopheles stevensii, which is a species in East Africa and in South Asia, and then also in Anopheles albuminus, which is in Central America and some of the Caribbean islands. So we don't work on Anopheles gambii ourselves, but there's no reason why this technology couldn't work in that species. And there is actually a big project which is coming out of it's one of the universities in London, Imperial College, which is trying to use similar technology in Anopheles gambii for malaria control in West Africa.
Okay. Thank you, Nathan. The next question is for you also. It says, why not eggs test? I think the question is, why not test eggs for antibiotic resistance?
Okay, so this comes back to the questions about tetracycline and whether the use of tetracycline in the production of female mosquitoes in the United Kingdom might lead the males in Florida to spread antibiotic resistance bacteria. And again, this is something which the EPA has assessed in great detail. It's also been assessed by Florida state regulators and also in the past by the FDA when they looked at Oxytec's earlier version of the mosquito OX5138. And in all cases, the regulators have determined that there is negligible risk of this happening. And so regulators have not seen the need to ask for any tests. And we respond when the regulators will ask us to carry out any scientific data generation, any testing. And so if there was a question that was to come from a regulator, we would obviously respond to that request.
Thank you, Nathan.
But in this case, they've determined there's negligible risk of that happening and therefore no reason to do those tests.
Thank you, Nathan. The next question is for you too. It's a regulatory question, and it says, you can release 1.3 billion mosquitoes according to the test review and amendment document from the EPA. When do you get to release 1.3 billion versus 500 million?
Okay, I'm not entirely sure I'm following the question, but the EPA in their approval of this particular project authorized the release of a certain number of mosquitoes per acre per week. And the maximum number of male mosquitoes which could be released per acre per week was 20,000. In reality, we're likely to release far lower than that because we have effective mosquito suppression in Brazil with much lower numbers of mosquitoes per acre per week. So that's the maximum level that was permitted by EPA in the permit, and that applies to both Florida and Texas over a 2-year period. And so that's where some of those big numbers come from. As we've said last time, the trial plan for Florida is likely to be far shorter than that, probably about 28 weeks in total, and with substantially lower release numbers expected. But I know that we're going to go into much more detail about that and why the project's been designed in a certain way and what the questions are that we want to answer. And I know that's going to be covered in one of the upcoming webinars, I think in 2 weeks' time. So I don't want to go into too much detail about that right now.
Thank you, Nathan. We've got just about 5 minutes until 6 o'clock, and we do have some more questions to try to get through. The next one is also for you, Nathan. It is, your technology seems strong, supported by robust science and regulatory oversight. Can you name any possible danger to humans, flora, or fauna in our environment as a result of your technology? And if not, why are many people in the Keys resistant to your project?
Okay, well, thank you for that question. I think it's a great question. And it actually summarizes the way that the regulators have assessed this. Their conclusion is, after assessing a huge amount of science, that there is no danger to humans or to flora or fauna or anything else in the environment as a result of using this technology. Those are not my conclusions. Those are pretty much word for word what the EPA concluded when they assessed this. And I think the bigger danger is not anything to do with the mosquito, but the real danger right now is the diseases that this mosquito can transmit. There is a dengue outbreak in the Keys right now, unfortunately in Key Largo, and it seems like the number of cases are continuing to increase. And that's the real danger here, and that's what this project is focused on, helping to provide a new solution for. And it's more broadly what we care about at Oxytec, is really being able to provide a solution to very real diseases which are affecting people in a lot of places in the world.
Thank you, Nathan. The next question is for you also, and we'll try to get through these next couple quickly. It says, why did Dr. Gorman say you were allowed to have so many females? And from your EPA documents, only 500 eggs were tested for females. That doesn't seem like a statistically significant sample size given 500 million.
Okay. Again, I'm not quite sure what this question is referring to, because as I've said earlier, we are not allowed to release any females. And if a single female was to be released, we would have to notify the regulators and stop releases immediately. And we have never seen a female released, and we don't expect that to happen either. So I also don't quite know where this statement about EPA documents and 500 eggs tested for females come from. That's not a number that appears in our EPA documents. We've tested far greater numbers. We've also done a large-scale deployment in Brazil of 20 million males and never seen females there either. And we shared field data from Brazil with the EPA as well. And as I said, we also have ongoing quality control to ensure that we are never going to be able to release any female mosquitoes. So I don't have any concerns there as it relates to that question.
Thank you, Nathan. I'm hoping this webinar might allow us to go over by just a few minutes. We only have 2 questions remaining, so let's try. But the next one, Nathan, is for you. Oxytec claimed all of those papers you showed earlier were independent reviews, but the first name I looked at was Derek Memo. How is this independent? And just for background for people watching, Derek Memo used to work for Oxytec.
Okay, so I just want to explain a little bit what peer review means and how it works, because I think that'll help to answer this question. So when scientists publish data, they will write it up in a paper, they will submit it to a journal, and the journal will send that particular article and all the data that it contains to independent peer reviewers. And that just means other scientists, other academics who work in the same field and who understand how the experiments were done and can assess whether or not they were done correctly, whether the data is of good quality, and whether the conclusions are appropriate. And so if Oxytec has published a paper, on that paper will be the authors, which will generally be Oxytec staff, and therefore some Oxytec papers will have Derek Nemo's name on them. Many of them have Neil's name on them, because he's been at Oxytec for several years. And so that means that those are the scientists who carried out the work. but it does not mean that they have done the peer review. The peer review is always done by independent scientists who have nothing to do with the experiments, who are independent at other institutions and can assess the quality of that work. And then typically what will happen is when the peer reviewers have looked at the work, they will determine whether or not the work is of good quality. If there are any problems with it, they will suggest that the scientists writing the article carry out additional experiments or reanalyze the data or reassess their conclusions so that the paper is scientifically robust and sound. And then only after all of that has happened would a journal actually accept a paper for publication. So that's how the process works. And so definitely some Oxytec papers would have Derek Nemo's name on them because he was a scientist who worked at Oxytec for many years and contributed to some of the science that was done at the company.
Thank you, Nathan. It looks like the webinar is allowing us to go over, so we'll continue if we can. But I mean, we'll wrap this up quickly. But these last two questions, they are also for you, Nathan. The first is, how do you know this technology won't affect other insects and also animals that may feed on them?
Okay, it's a good question. So it won't affect other insects because this is a species-specific technology that relies on mating. And so Aedes aegypti can only mate effectively with other Aedes aegypti mosquitoes. It cannot produce offspring with any other mosquitoes or any other insects. And so it's then only able to pass on the self-limiting gene to other Aedes aegypti mosquitoes. And therefore only they will be able to be affected by it. And the females will die and the males will be able to survive and pass on that gene, as we've talked about. Animals that feed on mosquitoes. So this is something which has been assessed in great detail. As I said, independent scientists did experiments to look at some animals that feed on mosquitoes, things like fish and other aquatic invertebrates that could feed on mosquito larvae. And there we showed, the other scientists showed, and we presented those data to the EPA, that the mosquitoes were not toxic and didn't have any impact on those animals that ate them. And so that is something that has been very carefully looked at as well.
Great. Okay, and then Nathan, do we believe that a 2-page marketing memo and an environmental assessment is sufficient for this trial?
Okay, I'm not quite sure what the 2-page marketing memo refers to. I'm guessing that it refers to a 2-page summary of the technology that the EPA provided as part of the information provided for the public comment period last September. That is certainly not what the EPA assessed when it was actually looking at all of the data provided. The EPA, as I said, assessed well over 4,000 pages of data, experiments, some from Oxytec, some from independent scientists, some from the scientific literature in carrying out its risk assessments for this project. That particular 2-page document was provided by the EPA to give a little more information to the public about the mosquito and how it works. And the EPA complied with all of the federal regulations that were required in terms of the amount of information provided for the public comment period. The EPA carried out an environmental assessment as part of its assessment of this particular project. They also looked at risks to human health, risks to animals, and so on. And obviously with all of that concluded no risk to humans or the environment. But that is all part of the EPA's requirements under the act which governs this kind of product and this kind of project. So the EPA complied with all of the requirements of that act in federal regulations.
Thank you very much, Nathan, and thank you, Neil. We had another question come in a few minutes ago, but I can answer this one. It's, can I speak to a scientist at Oxytec to learn more? And yes, absolutely. That's part of what our scientists are here for. And they have had those types of conversations, including with Florida Keys residents who are just interested and want to learn more. So please reach out to us. You've got this florida@oxytec.com email on the screen. We will be very responsive there. And we invite you to join us for our webinars over the next 3 weeks, and just really appreciate your time and attention and your interest this evening. Recording will be made publicly available. I think we'll have it up tomorrow, and we will publish a Q&A. And yeah, again, we invite you to join us for our future public educational webinars.